Good morning, everyone. It's our great pleasure to have Dr. Scott Fraser as our first keynote speaker. When we were putting the program together, we had a concern that people might not be here early enough because the traffic and stuff. But then it occurred to us, if we have Scott as the first speaker, then we are very sure everybody will be here on time because Scott always has the most beautiful talk with all the beautiful images and movies. As you all know, Scott is the director of the Beckman Institute Biological Imaging Center at Caltech, where interdisciplinary research is being done to use the most advanced imaging technology to follow events within the organisms. In other words, they think seeing is believing to study biological science. And the research in his lab range from developmental biology to genetics to neuroscience, and they use models such as zebrafish, chick, quail embryos to mouse and stem cells. I think today he's going to share his visionary view with us on how imaging technology can be utilized to study stem cells and regenerative medicine. Welcome, Scott.
Well, it's a real pleasure to be here, and I'm very excited about the work we've done thus far with Children's Hospital, and I'm looking forward to doing more. So what I want to share with you is a couple vignettes of what's happening in the lab, and make an argument that now is the time that we need to move from just sort of trying to see things at the cartoon level, or at the what happens on average, to really capturing what happens on a cell-by-cell basis as gastrulation takes place, or as a heart is built, and that unfortunately that's sort of disappearing into the light, or as a face is built in a developing embryo. And we've used embryos for a long time, not just because of the health impact, as David was bringing up, but for us as biologists trying to capture how a gene regulatory network executes biological form, embryos are the natural. It does it in a relatively small package, it does it quickly enough, and it exercises so much of its genome in a short amount of time that there's a chance of capturing the molecular dynamics that subserve the cellular and tissue level dynamics. We've been working for quite some time on the toolkit that we need for this, and some of those are novel transgenesis techniques or novel imaging techniques, and what I want to talk about at the end is the sort of molecular imaging techniques we're going to need to be able to go to harvest the variability that we see. So rather than cursing variability, we want to exploit it. So to try to get an ability to follow developing embryos, it meant that we had to be able to ask several questions at a time of the cells. And so Rusty Lansford and a fellow from JPL, Greg Behrman, and I, a few years ago developed something that became the new generation of Zeiss microscopes, multispectral laser scanning microscopes, that allow us to label and follow cells and actually see, I hope you can see all the little protrusions reaching out of the cell, so we understand the range of contacts of the cells, and also be able to label structures within them and exploit that ability to label structures to target a number of fluorescent proteins to do multiplex reporter genes and things of that sort. Others have used these sorts of technologies to great advantage. So the Borisy lab, now at Marine Biological Lab in Woods Hole, has been using the next generation of these scopes, the laser LSM 710 from Zeiss, to follow up to 15 different labels now, and by multiplexing coding, he's following hundreds of different bacterial communities and looking in at the bacteria within all of our mouths right now to understand the communities at the macro and the micro scale and the dialogues that are going on between those communities, both in health and in disease. So we think that multiplexing and molecular imaging at this level allows us to ask a lot of questions, but the thing that it's given us as well is a lot of headaches. And the headache is that as we've been doing this intravital imaging, what we find is that the embryos very seldom have read the primary literature. They don't know that all the cells are supposed to go from one place to another. They don't know they're supposed to have single origins of individual structures. And I think it's no exaggeration to say that every structure we've looked at, we've seen cells either leaving it that we didn't expect or contributing to it that we didn't expect, which means we underestimated the cellular and molecular dialogue that goes into building a tissue. Oh, this is great. Either it just got darker or, oh, anyway, okay. So, so what we need to do then is have a set of tools that let us really ask questions of the single cells and the cells as they cooperate together. Keeping in mind that gene regulatory networks themselves work in single cells, and that most of our assays involve some sort of indirect measure or a homogenate that brings together 100 million cells. And it's impossible then to assess the individual gene regulatory network. For example, if you homogenize me and David together, it's a very scary thought. So imagine that you were doing 100 million told that way, and you wouldn't understand either one of our pathologies or personality traits. Anyway, so what we really need are techniques that let us exploit this. That means we need to be down at single cell grain of analysis. We need to be able to see what co-varies. So that means that we have to space the embryo and stem cells and regenerating tissues or whichever area we choose, knowing that we want to take the assay to the tissues as they're doing their development or as they're doing their regeneration, rather than excising a few cells or homogenizing a bunch of cells and taking those to the assay. It means we've got some real challenges. None of these were built with an evolutionary constraint for imaging being important for their survival. And the cells are packed and moved and everything else. So I just want to show you one of the ways that we've been trying to get around some of these problems. And that is to try to make machines that let us follow a whole tissue. In this case, it's a Drosophila embryo. We use those because they're more sensitive than human or other vertebrate cells to light. So if we can develop a technique that lets us watch each one of the cells in this embryo as it develops, we have a chance of applying that to intact tissues in a vertebrate system. When we think about what we need for this sort of compromise between resolution and contrast and such, what we want to be able to do is put down the life history of every one of the cells. And this is showing you the end of the trajectory of all the development, all the motions, all the lineages and all that went into building this embryo. So we need to be able to follow those cells often enough that we don't get confused. So, all of us have traits that if we milled around the room, I could follow us all because David would be wearing his hat and Cambrian would be wearing his sweatshirt. But if I had no way of following them by those unique traits, I would have to reacquire an image of David and Cambrian before they moved half the distance between each other. So that sets a temporal standard that if we don't want to be confused, we have to meet. The problem is that as we image more and more, we use more and more light, and we end up perturbing the embryo as a result, or the tissue, even before bleaching appears. So to try to get around this problem, we've been building more and more expensive and more capable microscopes